Hello, my name is Luke Nunnery, and today I'll be explaining genetic regulation. Gene regulation is a process in which a cell determines which genes are expressed. We commonly see the selection of genes to make functional units, such as proteins. The genes expressed from a cell allows for a specific group of functional RNAs and proteins. Typically, genes are regulated at the transcription phase, but can be regulated at any of the many steps in gene expression. Cells in eukaryotes all contain the same DNA and many different genes. The ability of these cells to turn on specific genes to make a cell specialize is the purpose of gene regulation. An example would be a specialized liver cell expressing genes that coded for production of enzymes to detoxify the body. The liver doesn't need to produce insulin, so the genes that code for insulin production are turned off. This would be compared to them being expressed in the pancreas. Pre-mRNA is an immature molecule that isn't quite considered an mRNA molecule. Pre-mRNA must undergo modifications such as capping, splicing, and the addition of a poly A tail before the molecule becomes a messenger RNA. All of these modifications could be altered to result in a different protein or product than desired. In post-pre-mRNA formation, regulatory mechanisms can affect proteins that have already been made by the cell. We may see an addition of a chemical group like methyl or removal of an amino acid. These modifications after translation can edit or change the product of a protein. In the example of polycystic kidney disease in mice, alternative splicing could have taken place before the formation of pre-mRNA. Alternative splicing is the removal of introns and exons, which could lead to the coding of protein F. Post-translation, we might see the removal of an amino acid to produce protein F, as it would have coded for, say, a protein G before the removal of that amino acid. These are my citations. Thank you.